Hello and welcome to the Neuroanatomy Prelab 3. This is going to be brainstem and cerebellum. First, we're going to be looking at gross objectives. On the brainstem, we're going to have three main segments. The first is going to be our midbrain, which is going to be most superior, outlined here in blue. Inferior to that is going to be our pons, outlined here in green. And inferior to that is going to be our medulla, here outlined in yellow. First, looking at the medulla oblongata, this is an anterior view. The first most prominent structure that we see, most anterior and most medial, is going to be our medullary pyramids. Here we have our right, our left, and then on either side of that, just lateral, also a very prominent bump is going to be our medullary olive. The pyramids are going to hold our corticospinal tract, while our medullary olives are going to be home for our inferior olivary nucleus. Looking at the posterior view now of the medulla oblongata, we can see the more lateral prominent structures are going to be our cuneate tubercles. These are going to hold our cuneate tracts. Medial to that is going to be our gracile tubercles, which are going to hold our gracile tract. We also have our floor of our fourth ventricle. Next, we're going to look at our pons. This is going to be an anterior and sagittal view. The most prominent structure on our anterior side is going to be our basis pontis, which can be seen here on our sagittal view. Just deep to that and more posterior is going to be our tegmentum. And then our fourth ventricle is going to sit just deep to that, more posterior, in between our tegmentum and our cerebellum. On cross-section, we can clearly see our basal pons, which is going to be the most substantial part of our pons. Our tegmentum, again, is going to sit more posteriorly. Our fourth ventricle, sitting here. And then our tectum, which is going to be sitting here in blue. Pons, we're going to look at our posterior view now. The most prominent feature here is going to be our middle cerebellar peduncle, outlined here in blue. Next is going to be our facial colliculus. Remember that this is going to be our abducens nucleus with our facial nerve actually wrapping around inside of our pons, which is making this protrusion. Our vestibular area is going to be lateral and slightly inferior to our facial colliculus, separated by our sulcus limitans. Midbrain, this is going to be an anterior and sagittal view again. These prominent structures on the anterior surface are going to be our cerebral peduncles. And then in between our cerebral peduncles is going to be our interpeduncular fossa. This bright yellow structure that has just been brought up is going to be our tectum, which is going to hold our superior and inferior colliculi. Midbrain from a sagittal view, we have our superior colliculus. Again, is going to sit within our tectum. Our inferior colliculus, cerebellum, is going to be next. This is going to be a superior view. Two prominent structures that we can see here are going to be our anterior lobe. Just below that is going to be our posterior lobe, quite a bit larger, and that's going to actually be separated by our primary fissure, which sits here. Our horizontal fissure, which is located down here, is going to actually be separating our posterior lobe. Our vermis is going to sit right in the middle of our cerebellum, and that's actually going to split our cerebellum into two distinct hemispheres, the left and our right hemisphere. Cerebellum from an anterior view, we can see our nodule here, our flocculus out here. Together, these make up our floccular nodular node. Just beneath that on this illustration, we can see a protrusion out of our cerebellum. This is going to be our cerebellar tonsil. Our floccular nodular lobe is separated by our posterior lobe by our posterolateral fissure. This space is going to be our fourth ventricle. Cerebellum in cross-section, which is on a sagittal view, we can clearly see these finger-like projections, which is going to be our folia. Sitting in between the folia is going to be white matter called the arbor vitae, or the tree of life. And then we have our cerebellar cortex, which is the gray matter, which is going to sit outside of this white matter. Here. Again, a sagittal view. We can clearly see the fourth ventricle sitting in between the pons and our cerebellum.
Welcome to the second video for the Brainstem Lab. This portion focusing on the cross-sectional images of various brainstem regions. In this video, we will use information learned in the gross video to identify which level of the brainstem we are in, and which other structures should be located at that same level. Just like gross anatomy, positional relationships are very important to identifying structures in the brainstem. In this lab, we will focus on 8 brainstem cross-sections. We will start first at the level of the motor decussation of the medulla and work our way superiorly into the midbrain. This level of the brainstem exemplifies the transition between brainstem and spinal cord. Looking to the guides on the right, we can see from the posterior aspect that this section is below the gracile and cuneate nuclei, found here. And from the anterior aspect, this section is below the pyramids, seen here. As you should remember from the spinal cord lab, when evaluating structures on cross-section, tracts of myelinated axons will appear darker than collections of cell bodies. So when you see a darker structure, think white matter. Lighter structure, think gray matter. When viewing these images, the upper portion of the image is posterior. And here, separated by the posterior median sulcus, are the dorsal column tracts, just as they are found in the spinal cord. Each dorsal column is separated into 2 halves, the medial half being the fasciculus gracilis, carrying sensory fibers for proprioception, discriminative touch, and vibratory sense from the lower extremity, and the lateral half being the fasciculus cuneatus, carrying the same sensory information for the upper extremity. Sitting anterior to this, we can almost make out the remnants of an H or butterfly-shaped gray matter that we saw in the spinal cord. We can see these posterior horns, and there are some remnants of anterior horns. If we move down into the thoracic region of the spinal cord, we can see a very nice H-shaped gray matter surrounded by white matter. Within the white matter, remember our descending motor tracts, the lateral and anterior corticospinal tracts. The anterior tract is made of pyramidal fibers that do not decussate in the medulla, while the lateral fibers, the majority, do decussate. Remember from the external portion of this lab that higher in the medulla, descending pyramidal fibers run in the anteromedial enlargements, called the pyramids. Here at the motor decussation of the medulla, we can see where pyramidal fibers either cross to the contralateral side to become that lateral corticospinal tracts, or if they remain ipsilateral as they descend, they would be in the perfect position to become the anterior corticospinal tracts. Now, coming from the remnants of our posterior horn, what is really a continuation of the substantia gelatinosa is the spinal tract and nucleus of the trigeminal nerve. This structure carries pain and temperature fibers from the head and face. The substantia gelatinosa does the same for the body. With this structure, you will always see a mix of tract and nucleus together. Think yin and yang. This is due to the fact that the trigeminal nerve fibers enter and exit the pons, but the spinal nucleus of the trigeminal nerve travels all the way down into the inferior medulla. So there will be fibers descending from the pons all the way down into the medulla to relay in the nucleus. It's the descending fibers that form the tract, and the darker staining tract will always be seen lateral to the lighter staining nucleus. Moving anterior-lateral to the spinal trigeminal nuclei and tracts are the spinocerebellar tracts, responsible for carrying proprioceptive information from the extremities and ascending to the cerebellum. Medial to the anterior spinocerebellar tracts are the spinothalamic tracts, carrying pain, temperature, and crude touch from the contralateral body. In our second section, we'll look at the sensory decussation in the medulla. In this level, we can again find our spinal trigeminal nuclei and tracts. Anterior and lateral to those, we find our spinocerebellar tracts. Medial to the spinocerebellar tracts are the spinothalamics. The most anterior medial structures are the descending corticospinal fibers, which form the external medullary pyramids just before they cross at the motor decussation seen previously. In the posterior portion is where we begin to see big differences. We still have the cuneate and gracile tracts, but these primary neurons are reaching their first synapse at the respective cuneate and gracile nuclei. There is also another small nucleus lateral to the cuneate nucleus called the accessory cuneate nucleus. These nuclei receive some of the fibers from the cuneate tract and project to the cerebellum. Focusing on the gracile and cuneate nuclei, let's look at why this level is called the sensory decussation. First-order neurons ascend in the gracile and cuneate fasciculi to this level and synapse in their respective nuclei. From here, second-order neurons leave the nuclei, travel anterior and medial through the medulla, and cross midline. These fibers are known as internal arcuate fibers. The fibers cross to the contralateral side and ascend in the midline position just posterior to the corticospinal tracts. This ascending tract is termed the medial lemniscus. Next, we look at the level of the inferior olivary nucleus. As we move superior slightly in the medulla, we can review the structures we've already seen that are continuing here. We have the posteriorly located dorsal column tracts with the associated gracile and cuneate nuclei. Secondary neurons then decussate and ascend in the medial lemniscus. Lateral to the cuneate nuclei are the accessory cuneate nuclei. Slightly anterior and lateral to the cuneate nuclei is the spinal nucleus and tract of the trigeminal nerve. Remembering, the darker portion is the tract and the lighter, more medial portion is nucleus. Lateral to the spinal nucleus and tract, we again see our anterior and posterior spinocerebellar tracts. Medial to our spinocerebellar tracts, we again see our spinothalamic tracts. And lastly, the most anterior medial structures, the corticospinal tracts, which are above the level of the decussation, so they have not yet crossed midline as they descend. Next, looking lateral to the medial lemniscus, we see our inferior olivary nuclei for the first time. These have a very distinct tapeworm-like appearance. They are also so large that they produce a bulge on the anterior medulla called the olive, seen lateral to the pyramids. Moving immediately posterior from the medial lemniscus, we have a small tectospinal tract, which is involved in the coordination of head and eye movements. Immediately posterior to the tectospinal tract is the medial longitudinal fasciculus, whose functions include coordinating eye movement and sight and sound reflexes, to name a few. Moving posterior to the medial longitudinal fasciculus, we have the first cranial nerve motor nucleus that we have encountered. These are the hypoglossal nuclei, providing motor innervation to the tongue. We can actually follow the nerve fibers as they exit the nucleus, pass anteriorly, and then turn laterally between the olive and the pyramid. This is the point where we would find the hypoglossal nerve externally. Lateral to the hypoglossal nuclei, we see another motor nuclei for a cranial nerve, the dorsal motor nucleus of the vagus. Again, we can follow the nerve fibers as they pass anterior and laterally, this time posterior to the olive. Looking at the hypoglossal and vagal nuclei here, note their positioning. This is actually at the very inferior end of the fourth ventricle, as it's starting to shrink down to what will become the central canal of the spinal cord. Think of the two bulges created by these nuclei into the floor of the fourth ventricle, seen on the gross specimen. Posterior lateral to these motor nuclei, we find the solitary nucleus, which has multiple functions, one of which is the sensory nucleus for taste. One last small nuclei to identify, which is embedded within the reticular formation, is the nucleus ambiguus. You will find it posterior to the inferior olivary nucleus in this section. This nucleus gives off motor fibers that form the glossopharyngeal nerve and some motor output to the vagus. Next, we will move further superior to the level of the pontomedullary junction. At this level, we see our first clear opening of the brainstem posteriorly into the fourth ventricle. The structure protruding into that space from a posterior direction is the first time we see cerebellum in cross-section. This is the midline portion of the cerebellum, so it's specifically the vermis. Now, looking at the brainstem matter itself, we use our obvious inferior olivary nuclei to indicate that we are still near or in the medulla. We are now above the sensory decussation, so we lose our posterior column tracts and nuclei, but their secondary neurons continue at this level, medial to the olivary nuclei as our medial lemniscus. Anterior to these structures, we still see our corticospinal tracts. Looking posterior and lateral to the olive is the spinothalamic tract, and still in a similar location to previous slides, located posterolaterally, we can see the dark light mix of the spinal nucleus and tract of the trigeminal nerve. Posterior medial to this nucleus, brought closer with the absence of the cuneate nuclei, we again see our solitary nucleus and tract. Now that we are above the hypoglossal nuclei, the most posterior medial structure is the medial longitudinal fasciculus. Just anterior to it, as seen in the previous section, is the tectospinal tract. The first new structures we will identify at this level are located between the inferior olivary nucleus and the spinothalamic tracts. These are the central tegmental tracts, which carry ascending and descending fibers. At the level of the pontomedullary junction, we can easily identify the inferior cerebellar peduncles. Our spinocerebellar tracts reach the cerebellum through these peduncles, along with a few other cerebellar tracts. Around the inferior cerebellar peduncle, we also find the nuclei associated with the eighth cranial nerve. Posterior and medial to the inferior cerebellar peduncle, protruding into the fourth ventricle, are the various vestibular nuclei. This is seen as the vestibular area of the floor of the fourth ventricle. Wrapping around the inferior cerebellar peduncle laterally, we can identify the cochlear nuclei. Now we'll look at our first section of the pons at the level of the facial colliculus. Using our guides on the right, you should be able to see that from the posterior aspect, we are looking at a section through the facial colliculus, just above the stria medullaris, separating the pons and medulla. Before we get to naming tracts, we can separate the pons into a couple basic regions: the more posterior tegmentum and the anterior basilar pons, or basis pontis. Within the basilar pons, we have our descending corticospinal tracts and a bunch of small nuclei referred to as pontine nuclei. These nuclei give rise to transverse fibers, or pontocerebellar fibers, which cross midline and enter the cerebellum through the middle cerebellar peduncle. This is an important part of the cortico-pontocerebellar pathway. Moving on, we can again see the posterior opening of the pons into the fourth ventricle. The protrusions seen here are called the facial colliculi. These are so named because they are formed of facial nerve fibers protruding into the floor of the fourth ventricle. These can be seen arising from the lightly stained circular area found laterally in the middle of the tegmentum. This is the facial motor nucleus. The facial nerve fibers wrap around another cranial nerve nucleus, the abducent nucleus. You can follow the nerves exiting the medial portion of the abducent nucleus, running almost directly anterior to exit the pons just lateral to the pyramidal tracts at the inferior pontine sulcus. The facial nerve exits the pons more laterally in the same sulcus. To highlight tracts we have seen in previous areas, let's begin by looking in the floor of the fourth ventricle. From the external view, lateral to the facial colliculus lies the vestibular area. Deep to this surface is the vestibular nucleus. Anterior to the vestibular nucleus, we again have our spinal nucleus and tract of the trigeminal nerve. Medial to the facial nucleus is our central tegmental tract, and anterior to the facial nucleus is our spinothalamic tract. Again, looking to the floor of the fourth ventricle, the most medial structure here is the medial longitudinal fasciculus, with the tectospinal tract anterior to it. Moving further anterior, at the most anterior point of the tegmentum, we find our large medial lemniscus. Notice how it's starting to curve from its previous vertical shape to a more horizontal shape. Moving anterior to the medial lemnisci, in the most posterior portion of the basis pontis, lies the trapezoid body. This body is made of nerves from the cochlear nuclei, seen in the previous slide, crossing midline to ascend in the lateral lemniscus. Moving one step higher in the pons, we get to the level of the trigeminal motor nucleus. Here we are beginning to ascend to a level in which the fourth ventricle gets smaller once again. We know we are still in the pons by the obvious presence of transverse fibers in the basilar pons, which travel from pontine nuclei to the cerebellum through the middle cerebellar peduncle. Also in the basilar pons, you can see our descending corticospinal tracts, having not yet decussated at this level. The most medial, posterior structure in the tegmentum is again the medial longitudinal fasciculus, with the tectospinal tract just anterior. The medial lemniscus is beginning to move laterally from its previous position directly anterior to the tectospinal tract. Lateral to the medial lemniscus is the spinothalamic tract. Posterior to the medial lemniscus is the central tegmental tract. And lateral to the central tegmental tract is the lateral lemniscus. At this level in the tegmentum, we find a new group of nuclei, all related to the trigeminal nerve. The most medial is the motor nucleus of the trigeminal nerve. This nucleus provides motor output to the muscles of mastication. The most lateral of the group is the chief sensory nucleus of the trigeminal nerve, which is responsible for fine touch, proprioception, and vibratory sense from the head. Finally, the nucleus between the two and slightly posterior is the mesencephalic nucleus and tract. This nucleus receives proprioceptive information from the muscle spindles of the muscles of mastication. The final structures of interest in this section are the most posterior structures, found posterior laterally to the fourth ventricle. These are the superior cerebellar peduncles, containing tracts running between the cerebellum and the midbrain. Moving up the brainstem, we next encounter the midbrain, the first section of which will be at the level of the inferior colliculus. Looking at our guide brainstems on the right, we can see the inferior colliculi on the posterior view. This is the level of this section. Again, we can divide this portion of the brainstem, the midbrain, into separate gross regions. We use the cerebral aqueduct as our landmark. This is the narrow connection between the third and fourth ventricles. The region posterior to this is called the tectum. The most anterior lateral structures, the darkest staining structures, are the crus cerebri. These house descending tracts of motor neurons. The middle 3/5 contain the corticospinal and corticobulbar fibers, while the lateral and medial 1/5 contain corticopontine fibers. The space in between the tectum and crus is the tegmentum. When discussing all of the area anterior to the cerebral aqueduct, including the tegmentum and crus, this is called the cerebral peduncle. When looking at specific tracts and nuclei on this level, let's start with the structures that define this level, the inferior colliculi. Since these are involved with auditory reflexes, it makes sense that the lateral lemnisci would synapse here. The lateral lemnisci are ascending fibers from the cochlear nuclei. Anterior medially to these colliculi, we find the periaqueductal gray matter, which surrounds the cerebral aqueduct. Lateral to the periaqueductal gray matter, we again see nuclei of the trigeminal nerve, specifically the mesencephalic nuclei. These are the most superior nuclei of the trigeminal nerve. Moving anterior to the periaqueductal gray, we find two small nuclei near midline. These are the trochlear nuclei. This cranial nerve nucleus is the only one to send its fibers exiting the brainstem posteriorly, where they exit between the inferior colliculi, decussate, and wrap back to the anterior midbrain. Directly anterior to the trochlear nucleus is the medial longitudinal fasciculus. Anterior to that, as before, is the tectospinal tract. Lateral to these tracts is the central tegmental tract. At this level, the medial lemniscus has moved even more laterally and has started to curve more. Remember, these are second-order neurons from the dorsal column nuclei of the spinal cord ascending to the thalamus. Posterior to the medial lemniscus, we find the spinothalamic tract. The most central, dark structure is the decussating fibers of the superior cerebellar peduncles. Moving anterior laterally from this structure, you'll find two stripes of lighter colored matter. These are the substantia nigra. They contain important dopaminergic neurons for the basal ganglia. And don't forget our crus cerebri. The crus can be used to differentiate the levels of the inferior and superior colliculi. At this level, the crus are much more spread out, or defasciculated, as some of these fibers are prepared to interact with pontine nuclei. We've finally reached our last brainstem section. Starting where we finished the last slice, notice how much more compact the crus cerebri are at this level. Just posterior medial to the crus, we again have a pale stripe of dopaminergic neurons, the substantia nigra. Posterior medial to the substantia nigra, we have our first new structures of this level, the red nuclei. The red nuclei send descending motor fibers to upper limb flexors through the rubrospinal tract. If you remember from the spinal cord, this tract runs very closely with the anterior lateral system and is found near the spinothalamics in the brainstem. Moving laterally from the red nucleus, we can again find the medial lemniscus, still moving more lateral and posterior as we travel superior. And posterior to that is the spinothalamic tract. Now let's look at structures surrounding the periaqueductal gray, which still surrounds the cerebral aqueduct. At the level of the inferior colliculus, the most medial structure anterior to the periaqueductal gray was the trochlear nucleus. At this level, it is a pair of nuclei that provide preganglionic parasympathetic innervation to the eye, the Edinger-Westphal nuclei. Fibers from these nuclei join the oculomotor nerve and terminate on the ciliary ganglion. Anterolaterally from the Edinger-Westphal nuclei is another pair of nuclei associated with ocular musculature, the oculomotor nuclei. Coming from the Edinger-Westphal and oculomotor nuclei together, called the oculomotor complex, we can see oculomotor nerve fibers traveling through the tegmentum and exiting between the crusts in the interpeduncular fossa. Moving more anterolaterally from the oculomotor nucleus, we again find the medial longitudinal fasciculus. Looking laterally from here, we find the central tegmental tract. Posterolateral to the periaqueductal gray matter, we again find our mesencephalic nucleus of the trigeminal nerve. Finally, our last brainstem structure of interest, the superior colliculus. This structure receives visual fibers from the superior brachium and sends fibers via the tectobulbar and tectospinal tracts. It is involved in orienting movement of the head and eyes.
Moving on to cerebellum digital objectives now. Remember from gross objectives, we identified our folia, which is going to be our finger-like projections. Our white matter, which is going to be called the arbor vitae, or tree of life. And we have our cortex, cerebellar cortex here. Moving on to the deep cerebellar nuclei. Moving from most medial, we have our fastigial nucleus, globose nucleus, embuliform nucleus, and dentate nucleus most laterally. This can be remembered by the mnemonic, fat gerbils eat daily. Another illustration of this, we again can see our fastigial, globose, embuliform nucleus, and our dentate nucleus. At the pontomedullary junction in the open medulla, we can clearly see these prominent structures on the outside. These are going to be our inferior cerebellar peduncles. At the level of the facial colliculus, we can see our middle cerebellar peduncles, illustrated here. At the level of the trigeminal motor nucleus and the pons, we can see two prominent structures here. This first structure, outlined in orange, is going to be our middle cerebellar peduncle. Posterior is going to be our superior cerebellar peduncle.